welcome along. I'm very honoured to have been asked to offer some introductory remarks for this very special event. Most of you know me, and for those who don't, I'm Professor Karen Froud. I'm the Programme Director for Neuroscience and Education at Teachers College, Columbia University. And this event has been organised by our amazing student group, the Synapse. That group is led by current programme students Noam Eckhouse. Noam, say hello. Mahima Golani, who is right there. Say hi, Mahima. Sarah Bennett, Sarah. And most especially a recent graduate from our programme, Liri Haram. Say hi, Liri. This symposium was originally Liri's vision, and all of us are very happy to see it finally come to fruition. Liri has spoken very movingly about starting grad school and changing careers and countries during the pandemic, those feelings of fear and isolation, but also the opportunity that came with it to truly rely on and connect with peers and others. The Synapse events have been one of those anomalous pandemic wins, and we are all so grateful to have been beneficiaries of the connectedness and support that has come from those initiatives. So thank you all, and congratulations to all of you on such hard work and on the insightful and empathic approach that characterizes the work of the Synapse and that has engendered this timely and necessary discussion. Thank you and bravo. So everyone here has been impacted personally, professionally, emotionally, cognitively by the pandemic. Full disclosure, myself, I was acutely sick with the virus until just a few days ago, and I'm still quarantining at home and still feeling the effects of the infection. I can tell you that the brain fog is a very real thing. As a society, as well as individuals, no doubt we're going to be processing the effects of this seismic event for many years. And the impacts are being felt along two main fault lines: the effects of COVID-19, the disease itself, and the effects of pandemic-related isolation and the social change that we've all experienced. So today's symposium is structured according to those two frameworks. This morning's speakers will give us insights into how research is unraveling the effects of COVID-19 as a disease. And this afternoon's speakers will discuss the broader social and cognitive effects of the pandemic. With respect to the first approach, the SARS coronavirus has proven to be full of mysteries, in many ways being unlike some other viruses that we've encountered during human history. As researchers and scientists, we've been trying to determine on-the-fly responses to vitally important questions: how the virus impacts the immune system, why many patients have potentially deadly inflammatory reactions to the virus, what causes long COVID, why and how the virus interacts with certain underlying conditions to potentially deadly effect, especially diabetes, kidney disease and dialysis, obesity, hypertension, immune suppression, and why and how the virus has different effects at different ages and stages across the lifespan. An important direction for research has been how the virus interacts with different cells and systems in the brain and the central nervous system. So the pathophysiology and underlying neurobiology of COVID-19 as a disease, that's the focus for this morning's discussion. Before I introduce the speakers and the moderators for this morning, let me also tell you about the second approach, which is being addressed this afternoon. The coronavirus pandemic has significantly affected psychological well-being in much of the population. We know that cases of depression, anxiety, and loneliness are all on the rise. And while social distancing was a critical public health measure for the pandemic, a long history of research has really shown that social isolation can lead to cognitive decline. So the full scope of the underlying causal mechanisms of social isolation and loneliness in humans remains unclear, partly because we can't really study that with randomized controlled trials, right? It takes a very unusual circumstance to really be able to investigate that systematically. So this afternoon's panelists are going to discuss the severity and extent of the risks to social and cognitive health that are associated with the pandemic response, as well as some potential approaches to balance those competing risks. And that session will start at 3 p.m. So meanwhile, let's get started with this morning's panel. I'm very honored to introduce our panelists. Dr. Morgan Firestein is a research fellow in the Department of Psychiatry at Columbia University Medical Center. Dr. Firestein is part of a team collaborating across Columbia on the COVID-19 mother-baby outcomes, the COMBO study, that's helping us understand the complex and multifaceted effects of the COVID-19 pandemic on the health of the mother-child dyad. And in collaboration with the Departments of Obstetrics and Gynecology and Pediatrics, her research spans the prenatal through early childhood periods, And she works on identifying prenatal factors that contribute to neurodevelopmental disorders. She uses in vivo and in vitro methodologies and studies how the placenta adapts in response to maternal cues and how it acts as a protective barrier against maladaptive physiological processes in the mother. Her doctoral work won an award from the International Society for Developmental Psychobiology, and she's published widely, including in the American Journal of Obstetrics and Gynecology, Pediatric Research, the Journal of the American Medical Association, the Public Library of Sciences, and many more. Her research integrates psychiatry, obstetrics, and pediatrics to improve identification and early intervention for neuropsychiatric conditions. So welcome, Dr. Firestein. So give such applause and welcoming gestures as we're able through this, this medium. Dr. Joseph R. Fauver, I'm so sorry, I should have checked with you beforehand. How does Fauver sound? Feel free to jump in and correct me.
We pronounce it Fauver, but that's...
You say Fauver, okay, well you have a much more beautiful accent for that. So Dr. Fauver is an assistant professor of epidemiology at the University of Nebraska Medical Center. Dr. Fauver trained in microbiology at the University of Colorado and epidemiology at the Yale School of Public Health. Has worked on many viruses, West Nile virus, Zika virus, and has continued on to sequence the genome of the SARS-CoV-2 virus and to investigate the viral dynamics of COVID variants and how they interact with the vaccine. So really crucial and fascinating work. His work has been published in Cell, Public Library of Sciences Biology, Nature Microbiology, many others. And this research aims to use pathogen genomics to better understand infectious disease transmission and thereby to influence the public health responses. So welcome, Dr. Fauver. We're very glad to have you. And again, let's show such welcoming gestures as we're able. I know that for each of you, there would be a standing round of applause if we were able to be in person, but these are the limitations we're dealing with now. Finally, very, very glad to welcome Alissa Pacheco. I should have checked every pronunciation beforehand.
You had it.
I did. Thank you so much. We're very glad to welcome you as a TC alum. Oh, actually a current student, in fact. So licensed and board certified occupational therapist, working with individuals across the lifespan in a variety of acute care and inpatient and outpatient settings, including the Children's Specialized Hospital and the Atlantic Health System in New Jersey. Alissa's currently pursuing her doctoral degree here in motor learning and control at Teachers College. And she's exploring interventions for individuals with neurological and movement-related impairments. Alissa's been instrumental in adapting research paradigms to COVID conditions and has firsthand knowledge of pandemic effects on some of society's most vulnerable members, those with neurological disorders. And so to leave you, again, let's welcome our third panelist this morning. Thank you so much to all three of you for joining us. We're very excited for this, this first panel. Our moderators this morning are going to be two of our Synapse leaders, Sarah Bennett and Noam Eckhouse. And so I'm leaving you in very safe hands. Thank you for joining us and welcome.
Hello, hello. I think it's actually going to be me and Sarah. Hi, everybody. My name is Mahima.
And I beg your pardon, Mahima. Sorry about that.
No, all good. Thank you so much for that warm welcome into this space. And thank you to our panelists again for joining us. I'm really excited to dive in. I know we all are. Before we do, just a quick little background on the Synapse and kind of how it relates to this event. So a group of neuroscience and education students came together in the middle of the pandemic, feeling isolated from each other and from the world around us. And we started having these, these frequent conversations about like what to do to make it better. And we found that we all shared this passion for exploring and pursuing scientific knowledge, but also sharing it in an approachable, thought-provoking, and creative way. And from there, the Synapse was born. For our community, some quick reminders. If you would like to, please turn your video on. Seeing everyone smiling or deep in thought faces usually helps in these types of COVID events, these Zoom events. Also, we wanted to ask you to please stay muted during the event just to make sure things go smoothly. If you have any questions or comments or big reactions that you have to share, we can use the little functions that we have here, but also please feel free to use the chat. Specifically with questions, please add them there and we'll try to get through as many as we can in the time that we have today. And with that, I'll pass it on to Sarah and then we can jump in.
All right, thank you, Mahima. Thank you so much, Dr. Proud. And thank you to all of our panelists for being here and for all of you for joining us. We're so excited to have you. So for our speakers, we're really looking forward to having a conversation today and learning from your different perspectives and expertise in the different fields that you're in and even in your own personal experiences, of course. So before we move on to questions, I just want to remind the audience, as Mahima said, to submit any questions in the chat box. And I hope everyone is ready to dive in. Okay, so our first question is focused on COVID as a disease. And we want to start right at the beginning.
Sorry, honey. Before we start off, should we do a little intro for all the speakers to introduce themselves for a couple minutes?
Yes.
Yes, before we dive into anything else. I don't really know who goes first. Does anyone want to kind of start off and tell us a little bit about themselves, their paths, and their experience with COVID-19?
Hi, everyone. I'm happy to jump in. I have just a few slides. I definitely want to keep this discussion based, but maybe I can just give you a little bit of background on what I've been working on during the pandemic, and hopefully that will inspire some questions and thoughts. Let me just switch over my screen 1 second. Let's see. Okay, does this look correct? Okay, great. Well, first off, thank you so much for including me in this fantastic program today. I've really been looking forward to it. And I just want to briefly introduce our COVID-19 mother-baby outcome study. And really the focus that I'm going to, I guess, talk about today, a lot of the effort that I've been putting in is looking at these early neurodevelopmental risk assessments. Historically, a lot of my work has really been looking at these early child and parent interactions across infancy and early childhood. So, we know that the parent-infant relationship really develops over the first few years of childhood and is really critical in establishing a healthy, dynamic between the two, but also especially when we think about longer-term child development. But of course, many of these processes actually begin much, much earlier during pregnancy. And we now really recognize that the roots of the parent-infant relationship and many different infant developmental milestones and processes emerge earlier in utero. And this concept is supported by a framework that many of you may be familiar with. We refer to it as the Developmental Origins of Health and Disease, or DOHAD. Which really suggests that the prenatal environment and the experiences during pregnancy shape the fetal brain and ultimately infant development. And there are two factors that are very often discussed in this literature, and they include exposure to viruses and other pathogens and toxicants, and then these sort of natural experiments of stress exposure. So many lessons can really be learned from previous experiences of both of these. I'll just highlight a couple quickly. Perhaps most famously, the Zika virus during pregnancy is associated with many different morbidities, but when we think of long-term outcomes, exposure during pregnancy is associated with language deficits in childhood. And then influenza, rubella, poliovirus, herpes virus, and HIV are all associated with later onset of psychotic disorders in adulthood. Relevant to SARS-CoV-2, which is I know the focus of today's program. Interestingly, the prior sort of related outbreaks of SARS-CoV-1 and MERS during pregnancy, there's very few documented cases of those pathogens during pregnancy. And so going into this pandemic, we had very little data on child outcomes following exposure to these types of coronaviruses. And then when we think of these natural experiments of stress exposure, there's a very long history of work looking at famines, natural disasters, periods of war, all being associated with risk for schizophrenia, a range of language and cognitive deficits, and difficult infant temperament, again, in those infants that were in utero during these periods of stress. And, you know, when the pandemic began, I think my colleagues and I all realized that there is no better example of these two types of factors converging than the COVID-19 pandemic. And so my colleagues and I in March 2020 began the prospective combo study, as we call it, to really track numerous maternal and infant outcomes. And I'm not going to go through all of the details, but our study enrolls prenatally through 24 months of age. We collect a range of survey-based data, biospecimens, video-based data. We're conducting MRI imaging on mother-infant dyads through the first year of life. We look at smell, maternal and infant smell. Of course, anosmia is one of the early behavioral indicators that emerged when we think of COVID-19. They can enter at any point during this study, and we currently have about 1,300 dyads that are participating in all or some of the components of the study. So I'm sorry, I'm probably running over, but let me just give a quick snapshot of some of the work that we've been really excited about. So at 6 months of age, we had parents complete a parent report questionnaire of child neurodevelopment. And this assesses these 5 primary domains of development. And we had about half of the infants who had been exposed in utero to SARS-CoV-2 and half who were unexposed. And we were thrilled to see that there were absolutely no differences whatsoever between these two groups of infants. However, when we collapsed these two groups, given that we now, we know that they're not developmentally different, and we compared them to a historical cohort of infants who were all born and assessed prior to the onset of the pandemic. So here, the infants who were born during the pandemic are shown in purple. The historical infants are shown in gray. What you can see is that the infants born during the pandemic, again, regardless of maternal SARS-CoV-2 infection, were scoring significantly lower on gross motor, fine motor, and personal social skills. Now, I just want to point out quickly that statistically, these are quite robust differences, but when we think of the effect size, it's not a particularly large effect size. But on the other hand, we're talking about hundreds of millions of babies that have now been born during this pandemic. So on a population level and from a public health perspective, this is something we really, really do need to be aware of. I'll just add that this is unpublished, but we just have analyzed some data from a new sort of subset of our sample. So the data that I just showed you, these are all babies in the pandemic cohort that were born or in utero during the first wave of the pandemic, so really the acute phase. We now have data from infants who were in utero during the second wave. And what is really encouraging is that we actually seem to be seeing a recovery. So the purple are the first wave, gray are the second wave. And so those scores seem to be sort of normalizing back up to where we would have expected relative to the pandemic. And the last thing that I'll just allude to is, of course, this is all parental report data, which is subject to reporting bias that I'd be more than happy to discuss today during our session. So we have simultaneously been conducting video-based observational and standardized assessments of almost the same developmental domains by trained staff. And just looking at those who were and were not exposed in utero, again, very encouraging. We're not seeing any differences between those two groups. So again, thank you so much for having me, and I'm really looking forward to our discussion today.
Oh my gosh, that was incredible. And it just makes me even more excited for all that we get to talk about today. Who wants to go next from our exciting panelists?
I can go. Okay, can everyone hear me all right? My video is a little bit breaking up. Okay. So I also prepared something, not slides, just my story. I have a little bit of a different perspective. It's actually from the patient side of the experience and my involvement as a patient with COVID and long COVID. So if you have a couple minutes, I'm just going to tell my story. My name is Alissa Pacheco. I'm currently in my 4th year of my doctoral program in motor learning and motor control at Teachers College. I have my master's in occupational therapy and have worked in neurorehabilitation for over 10 years. I'm not here today, though, to talk about my background in rehabilitation, rather my experience with COVID-19 and long COVID. In preparation for this talk, I looked back at my 141-page COVID journal to help gather my thoughts and prepare to tell my story. It was mid-March. The whole world had basically shut down. I was still working in the hospital and seeing patients as normal. My symptoms began on March 23rd with what I initially thought were allergy symptoms. A few days later, I developed low-grade fever, fatigue, weakness, terrible cough, and difficulty catching my breath while talking. All I wanted to do was sleep. I called my doctor, who ordered a COVID test, but it would take over a week to get tested and then wait for the results on top of that. They started me on antibiotics for pneumonia, as my doctor said she could tell even over the phone that I had some sort of pneumonia. The antibiotics, of course, did nothing, and I got progressively worse over the next few days, with fever progressing to 103, terrible cough, extreme pressure headache, shortness of breath, severe sore throat, and body aches. I was so fatigued that I could barely move or talk. I didn't even have the energy to lift my arms to eat or drink, read a book, or turn on the TV. I literally just laid there on my couch, trying to focus on breathing. I felt like I had to focus all of the little energy I had stored in taking every single breath. I couldn't even talk because it would take away from the energy I needed to breathe. I remember being afraid to fall asleep because I wouldn't be able to focus on breathing anymore. The pain in my rib cage and either side of my lungs was excruciating. The thought of food made me nauseous. My taste had become metallic and everything smelled like burning garbage. I lost so much weight, I was down to about 95 pounds. A nurse from my doctor's office checked in on me 3 times a day for weeks, and then weekly my doctor checked in on me for another 6 weeks. It took almost 3 weeks for my blood pressure cuff and pulse ox to arrive because everything was on back order. Luckily, I had the support and recommendations from therapy colleagues who reminded me of pulmonary rehab techniques such as postural draining and percussion, which for those of you who don't know is a form of tapping on the lungs to help break up the mucus. And then you go on in different positions to have gravity pull the mucus out of your lungs. I had my husband watch YouTube videos to learn how to perform percussion techniques and understand what postural draining was to help me try and clear my lungs. I never understood how uncomfortable postural draining was until I had to do it on myself. I was so weak that my husband had to use pillows to help place me in the different postural draining positions to let gravity drain the fluid through my body and out of my lungs. It would take less than 5 seconds before I would feel the extreme pain of fluid moving through my lungs. Then I would have a coughing fit where I felt like I was choking on my own mucus as it exited my body and came out in large amounts at a time. We did about 3 rounds of percussion and postural draining 3 times a day for about 10 days, each time coughing up extreme amounts of fluid. No matter how uncomfortable it was, though, I knew how important it was to get the fluid out of my lungs. So it became part of my daily routine. After about 2 weeks, the fluid started subsiding. I felt like I was through the worst. By the time my pulse ox arrived 3 weeks later, my oxygen was 93% and would drop into the high 80s when I tried to walk to the bathroom or across my house. I have no idea what it was in the first 3 weeks of my illness. My heart rate would climb into the 160s. I couldn't understand why I was so sick so many weeks later when all you heard all over the news was COVID only lasts 2 weeks and doesn't affect young and healthy people. Why was I so debilitated? I had so much difficulty trying to get back to basic household tasks. As an occupational therapist, I had worked with patients on activities of daily living for years. And now for the first time in my life, I realized I couldn't do the things I needed to do to care for myself. Weeks later, I was still sleeping almost 15 hours a day and didn't feel rested. My fevers lasted on and off for 6 months. I was getting rashes all over my body almost daily. My hair was falling out and I was having allergic reactions to so many foods that I had eaten for my entire life. My joints were on fire. I could barely lift my arms to get dressed. I was so weak. I kept trying to get my endurance back by going up and down our driveway, but my oxygen kept dropping into the low 90s and I was tachycardic. My BP would drop every time I tried to do the slightest bit of exercise. I would be exhausted for days just from one trial of yoga or one walk down the driveway. I was sent to the emergency room 3 times by my doctor to get cardiac and pulmonary tests and blood work done because no doctor would see me in person due to my continued fever. I was told to stay in quarantine from March 2020 through August 2020 because they didn't know if I was contagious or not. My voice had completely changed. I was still having trouble having conversations because I would get very short of breath and my voice would become raspy and my throat very sore. I was having debilitating brain fog and trouble concentrating. When I still wasn't better after about 3 months, my primary care physician started sending me to specialists. I went from a healthy and athletic 33-year-old to a homebound woman who could barely walk from her bedroom to the bathroom or kitchen without getting exhausted. I saw over 12 specialists the summer of 2020, all who saw me virtually and who followed me every month to try and figure out what was wrong with me. Everything was negative. I was given more medication than I knew how to handle. By this time, I was part of numerous online support groups for people who had had COVID. I knew that my symptoms were similar to others around the world who still were not better, but many doctors didn't believe this was from COVID. They all gave me different diagnoses. In August 2020, 5 months after my diagnosis, the first long COVID clinic opened up in the hospital system that I worked. He was the first doctor who saw me in person 5 months later. It was life-changing for me. I was told that my story and symptoms were very similar to others who had had COVID. The doctor spoke to me for over an hour and validated everything. Even my hair falling out, allergic reactions, and frequent fevers, they said, fit into other people's experiences and stories. I was enrolled in the long COVID program at the hospital, where they still follow me every 3 months to this day. My case study was even presented in an international COVID conference. They sent me to the appropriate specialists who all saw me in person and addressed many of my symptoms. I was diagnosed with sleep apnea and started on a CPAP machine, given a steroid inhaler to help my breathing, medication to increase my blood pressure, and antihistamines and mast cell stabilizers to control my rashes and allergic reactions. I saw a functional medicine doctor who started me on about 15 different supplements to help with cellular repair, inflammation, and detox. I was sent to an ear, nose, and throat doctor who finally looked in my throat, the first doctor to look in my throat in 5 months, and said, and it turns out I was having bleeding in my vocal cords from all the coughing I had experienced during COVID. There was scarring as well. I started physical therapy where I attended 3 times a week for 11 months and also saw speech therapy 2 times a week for 6 months to address my vocal cord issues. Slowly, I started feeling myself again. My experience with COVID has taught me so much. I learned how to stay motivated, positive, and take each day at a time. I learned that good days and not so good days are both normal and part of the process. I gave myself permission to heal. There was a determination in me that I didn't even know existed. I learned to advocate for myself and didn't stop searching for doctors until I was happy with the plan. I realized how important a good support system was and was fortunate to have an excellent one of family, friends, and colleagues surrounding me. I learned the value of support groups and had started attending them weekly. Just hearing other people's stories and their perseverance motivated me to keep going. I also learned the value of citizen science and had joined multiple research studies. It took over a year for me to go back to work in the hospital with patients, but I believe I came back a better occupational therapist than I was pre-COVID, because now I truly understood what it was like to be a patient. I understood that healing is a full-time job in itself. Spending your day going from doctor's visits to therapy appointments, handling insurance claims, scheduling appointments, completing home exercise programs, all while not feeling yourself, takes up so much time. And that was something that had never really occurred to me before. I also had a deeper understanding of the frustrations that patients can go through when people don't listen to them, rush through appointments, or act like they don't know their bodies the best. I also never understood how debilitating fatigue truly was, both physically and mentally. And I had worked with probably hundreds of people with fatigue. Our patients have been through so much, and we are really just seeing the tip of the iceberg within our own individual disciplines when we work with them. I now have worked with numerous people with long COVID. So I tell my story to some, others I don't. It really depends where they are in their own personal journey. Either way, I try my absolute best to truly listen to their story, look into their eyes, connect with them and their experience, so we can work as a team to figure out the best way to help them return to their prior level of function. Thank you for listening.
Thank you so much, Dr. Pacheco. We can see in the chat, I know you've been reading, but so many people are so grateful for you, for your vulnerability and sharing your powerful story with us. And I know Dr. Fauver was saying that it's hard to remember how little we knew about the virus at that point. And I just want to say too, that it's so powerful to see and inspiring to see how you've taken this into your life's work as an occupational therapist. And yeah, we just want to thank you so much for sharing and for being here. And we're really looking forward to talking more. Yeah. And all right, Dr. Fauver, do you want to close us out with the introduction?
Sure. Yeah, thank you for sharing. That was incredibly powerful and frankly difficult to listen to. I was in the New York area at the time when COVID hit, and I remember just how difficult that was for everyone, and we managed to avoid infection. So, thank you so much for sharing. So I wanted to give just a little bit of background on my work and how I've kind of approached this pandemic and the work that we've done on it since. And I do have some slides that I would like to share to kind of help kind of orient some folks. So neuroscience is definitely outside of my area of expertise, and I come at this much more from a molecular biology, evolutionary biology standpoint. But I think, as we've seen with SARS-CoV-2, there's so many different facets of this that it kind of takes all these different disciplines to really get a good grasp on what's going on here. So my background is actually in medical entomology and arboviruses, so mosquitoes transmitted, or diseases transmitted to humans by mosquitoes and ticks mostly. So I spent a lot of my time in graduate school at Colorado State working on West Nile virus surveillance, which is a really, really common mosquito-borne disease. That, from a neuroscientist standpoint, it's probably very interesting because that is one of the clinical manifestations. I come at it much more from the insect side of things. But oddly enough, in graduate school, I was a part of another pandemic of Zika virus. And again, thinking about all the different connections and how you can come at these new, well, Zika wasn't new, but it was definitely emerging. These new viruses coming in, into society and seeing what the effects are. So obviously Zika was super consequential for a lot of people, and something that unfortunately has kind of fallen to the wayside in our considerations of it. I do feel like as someone in the infectious disease space that kind of collectively, whether it be from just attention or where funding is dedicated in things that we kind of focus on in the infectious disease world, we just kind of hop from one pandemic to another, whether it's Ebola, then Zika, then SARS-CoV-2, and MERS was in there, influenza, now monkeypox. It does kind of feel like we're constantly chasing and never really up to where we need to be. And one thing I'm really interested in doing is trying to improve infectious disease surveillance systems and understanding of how these diseases move through populations. Again, kind of from the pathogen side of things. So I was actually at the Yale School of Public Health when COVID started. I had just started my second postdoc there and was really interested in dengue virus, another mosquito transmitted virus that's actually now made its way to the continental United States through dengue virus transmission in southern Florida, but it's typically the disease of the tropics. And then shortly thereafter, COVID hit, and the world kind of turned upside down for everyone. All of our work shifted to SARS-CoV-2 and our background and the way that we kind of approached this problem was if we can get the virus in our hands and we can do what's called next generation sequencing. I don't know how familiar folks in the audience may be with that, so I'll kind of give like a brief rundown of what we were doing with those techniques and how we could learn about the virus by sequencing the viral genome. And we've been fortunate to have really great collaborators, some at Columbia actually, to put out, I think, some really kind of important work characterizing not only new variants and what those new variants may mean for the pandemic, but also kind of individual time courses of infections through a pretty well traced cohort. So I'll actually share some slides. I wasn't planning on it, but I do kind of have like a general slide deck that gives a bit of an overview of how we kind of try to answer some of these questions. So I'll try to run through these pretty quickly. So this was a talk I gave a while ago. And I think this kind of captures the sentiment here. When we're thinking about different variants of the virus, I'm sure you've all have become familiar with this. You know, the first variant of concern was Alpha, the quote unquote UK variant. That kind of upended the pandemic, and then Delta upended the pandemic, and now Omicron has upended the pandemic yet again. So I like to show this slide to kind of bring the point home of why understanding different variants and the genomic sequence of those variants is important. So this is outdated. So this was the peak of the Omicron wave, so January-ish. So if you were to look at this now, you would see that it dropped and it's actually gone back up and plateaued a little bit since. But the question I always ask is what happened? You know, if we weren't understanding how the virus was changing and evolving through time, this epi curve, so this is just from the New York Times, so x-axis is date, y-axis is number of cases. This epi curve wouldn't make sense if we were dealing with the same virus that emerged in early 2020. So at this point in time, roughly 50% of the eligible population was vaccinated in the United States. And then we saw a really big increase last summer. And then, you know, we saw a big increase. At this point, like 75% of the eligible population was vaccinated, and you know, even more had been exposed to have some amount of pre-existing immunity to the virus. So why would we see these huge peaks in daily cases that we saw this winter? And when we ask the question, what happened? The answer is the virus evolved. The virus has changed. We got the Delta variant and then the Omicron variant, and now we're dealing with more subvariants of the Omicron variant. So this is just a frequency plot. So it's, again, through time, trying to model the number of SARS-CoV-2 cases, in this case in the United States, and what proportion of those cases can be attributable to any given variant. Just to break down a really, you know, I think on the surface, a simple question, but actually I think somewhat of a complicated question to answer is what is a SARS-CoV-2 variant? So I think we should back up a little bit and start off with what is SARS-CoV-2. It's an RNA virus. It's in the family Coronavirinae. We are familiar with other coronaviruses. As was mentioned earlier, you know, we've had to deal with the original SARS coronavirus that was circulating in the early 2000s. Had to deal with MERS since, the Middle East Respiratory Syndrome coronavirus, and now we are dealing with SARS-CoV-2. So these are all in the genus beta coronavirus. Basically, they're all kind of related to each other, but still pretty distinct things. You know, if we thought about this as like a family tree, these would be cousins. And then there's these other human coronaviruses that circulate in the population routinely. Actually, you know, about 25% of common colds or upper respiratory infections are caused by these other 4 human coronaviruses. So again, all similar, but you know, different in their own way. And as we've seen throughout this pandemic, even small changes to the viral genome can drastically affect, you know, how the pathologies can change in individuals, how the course of infections can change in individuals, and how infectious this virus actually is. So, because you know, this is what we deal with again is the viral genomes themselves. So this is a cartoon of what a SARS-CoV-2 virus looks like. You know, it's this little spiky piece of protein that has RNA as its genome. So, you know, everything has a genome, right? Humans have very large genomes, double-stranded DNA genomes that are in the range of billions. SARS-CoV-2 has this tiny 30,000 base pair genome that uses RNA instead of DNA. And actually, if we're thinking about other RNA viruses, so most pandemic viruses are actually caused by RNA viruses. As I say that, the next thing everyone's looking at is monkeypox, which is actually a double-stranded DNA virus. But Zika virus, Ebola virus, influenza virus, SARS-CoV-2, these are all RNA viruses. And the thing about RNA viruses, they mutate relatively quickly, especially on the scale of transmission events. So it's just 30,000 pieces of RNA that code for a variety of different proteins. And then the question is, how do we find new variants? You know, how do we know when this genome has changed, and if that change in the genome is actually significant? Well, we look. So we've been doing this now since really the beginning of the pandemic. The first case occurred in Connecticut in early March of 2020. We were actually able to get the clinical sample from those first 10 cases, sequence the genome, and I'll show you what that means in a second, and learn something about it. So we use this process called next generation sequencing, which is, it takes a sample. I'm sure as you've gone through this pandemic, many of you, I myself have been tested for COVID dozens of times at this point. So it's a sample collection, whether that be a saliva sample or a nasal pharyngeal swab, just like you'd be getting, the same sample being taken for tests can be used as input into learning about the genome. So the way a SARS-CoV-2 test works, at least for the not the rapid diagnostic test, but for the PCR molecular assays, is they collect this sample, then you do what's called RNA extraction, so just take all of the RNA out of that sample. You then run PCR to determine whether or not there's SARS-CoV-2 in that sample, and then you go through this process called library preparation, which is really just adding a little bit of molecular biology to add bits and pieces of DNA onto the end of your SARS-CoV-2 RNA, and then you sequence it. And that the simplest way to describe that is it's reading all of the A's, T's, C's, and G's in the SARS-CoV-2 genome as accurately as possible. I have U in parentheses because this is an RNA genome and it uses a different nucleotide than DNA. Then we create what's called a high quality consensus genome, which is to say, if I was infected with SARS-CoV-2, here's the virus that infected me. Here's all 30,000 base pairs of that virus. It's going to be slightly different than all of the other SARS-CoV-2 genomes. And this goes to show you how outdated my slides are. This is from a talk I gave in February. This number is almost doubled since. So there's over 10 million SARS-CoV-2 genomes generated worldwide. And there's ways to visualize that. And for folks that are not in this space, this is far and away the best sampled virus that there's ever been, and it's really not even close. There's probably, maybe I estimate this, of all other viruses combined, so HIV, influenza, Zika, Ebola, whatever, combined, there's probably about 1 million genomes generated. So SARS-CoV-2 has been sampled on a scale that no other virus has ever been sampled. Okay, so what are we actually looking at when we're thinking about variants? Really, it's this RNA genome plus some number of mutations that might confer some advantage. It might make the virus more transmissible. It might make the virus more immune evasive. It might help the virus get around, vaccine-induced antibodies, things along those lines. So here is the World Health Organization's definition of what a variant is, which is, I think, just a more wordy way of saying the same virus that's gathered mutations that has made that virus somehow better at infecting people. So we can actually track this over time. So there's this website, outbreak.info, if you're really interested in this. And all this is doing is plotting the different variants as they've occurred over time and what proportion of new cases are caused by any given variant. So if you all remember, in kind of the winter of 2020, first part of 2021, the Alpha variant, or the so-called UK variant, I'm very happy we've moved past naming variants after locations. We should be moving past naming any infectious disease after location because it is quite stigmatizing. But remember, the Alpha variant kind of won out. The Delta variant came in very quickly and was winning the day. And then I still remember I was at a Thanksgiving dinner with my family, and I looked at my phone, and there was this new post out of these incredible scientists in South Africa and Botswana describing this new variant that had all sorts of mutations in the spike gene. Spike gene is really important for the virus to get into cells. It's what the vaccine is made out of. That there's this new variant that has all these different mutations, and we think that this can be really consequential for the pandemic. And that's absolutely been the case. The number of SARS-CoV-2 cases since Omicron's emerged, and we're in the US alone, tens of millions since. So just really quickly, just to run through to give you a bit of the background about some of the stuff that we've been studying specifically, and hopefully this can help maybe orient some questions. We tend to ask these questions: why do we see some of these variants win out over others? Why are some better at infecting people or moving through populations or whatever? There's really two different ways. The virus can be more transmissible, inherently more transmissible. You could think of things like R0. That's a number that got thrown around really early in the pandemic. It's really the idea of if one person's infected, on average, how many more people would they infect? And there's a few different ways a virus can become more transmissible, or the virus can be more immune evasive. So, the virus that's circulating today is different than the virus that was circulating at the very beginning of the pandemic. And the vaccine, which is designed to that earlier version of the virus, these new variants, may have mutations that help get around human immune responses. So one way to track this is just by looking at proportions, as I've shown you previously. Another way to track this is by measuring just kind of increased transmissibility of any one variant. So these are our frequency plots that are showing what's called it's similar to R naught. It's this idea that number of transmissibility of any one variant kind of changes over time due to a variety of factors: the virus changing itself, the seroprevalence in the population, the vaccination rates in populations. Basically, to say it's going to go up and down and ebb and flow. But there's also ways that the virus in an individual infection, be thought of as an individual infection, can be more transmissible. So what I'm showing you here is kind of a cartoon of this paper that we published, showing you in an individual infection, on average, this changes drastically from person to person. What does an infection look like? So what we're measuring here on the x-axis is time, on the y-axis are CT values. So this is the output of a SARS-CoV-2 diagnostic assay. You can think of this as quantitative. So more virus as you go up, less virus as you go down. So over the course of an infection, if someone tests positive, they're going to have a little bit of virus, and they're going to have more virus, then a lot of virus, and then they're going to clear it eventually. And actually being able to quantify these things such as proliferation, peak CT value, and clearance rates gives us an idea of how these variants may behave differently in individuals. So how are we able to do this? Oddly enough, we have a really great collaboration with the National Basketball Association. From the very beginning of the pandemic, the NBA has been probably, if not the most well-tested cohort in the world, one of the most well-tested cohorts in the world. We started working with them when we developed the saliva-based diagnostic assay called Saliva Direct. Basically, instead of getting your brain poked with a swab, you can spit in a tube and detect SARS-CoV-2 quite readily. But after we worked with the NBA to help get a sample size to test this, whether or not this diagnostic test was useful or not, we started working with them to do kind of through their routine testing program, understanding some basic research questions about infections, because this cohort was testing every single day, which just does not happen very often. So we're able to ask questions about the new variant, Omicron, by these various factors. The peak viral concentration. So if you're infected with Omicron opposed to a previous variant, do you just have more virus in your system? Is that why that virus is more infectious? There's just more virus in someone for longer. And it turns out the answer is not really. Another question would be, if you get infected with the Omicron variant or any of the new circulating variants, do you get more virus in your system quicker? You can imagine if you had a lot of virus in your system before you're symptomatic, you're more likely to transmit that to someone else. Or maybe you're infected for longer. Maybe the clearance time is slower. So you have more virus in your system for a longer period of time, you're infectious for a longer period. And then overall, we can quantify kind of the acute infection duration. And what we learned is that for a lot of these variants, they don't really change much on an individual level. When we take the averages across these cohorts, they don't really change. But one thing we were able to determine readily is that being vaccinated actually reduces the time of your infection, which we think has been really advantageous. And that's, again, with some of these newer variants. And then so the last thing I wanted to show you is a preprint that we have out right now. This has been updated since, and we should have a new preprint out soon, but kind of thinking about how virus evolution can affect public health policy, how these changes and the virus should then correspond to the policies that we have in place. So folks may remember kind of in the middle of the Omicron wave, the CDC guidance on an isolation period. So once you tested positive for the virus, how long do you need to isolate for? Changed to 5 days. I was very outspoken about this. A number of my colleagues were very outspoken about this, that 5 days is just not long enough. And our cohort really shows that to be true. So what we're plotting here is the proportion of people that were from their first positive test. Folks were not testing every single day. It's when they felt symptoms, they took a test. At 5 days, how many of them still had, this is called a proportion of CT of virus in your system with a CT value less than 30, which, to simplify that, is after 5 days, how many people still had a lot of virus in their system that we would presume they are infectious? And the vast majority in the cohort at the 5-day isolation period still had very, very high detectable levels of virus in their system. So I hope that that kind of gives you a broad overview of where we're coming at this from, the sorts of questions that we can answer with these data, and really about how virus evolution should, we think, dictate public health policy. So thanks again for inviting me. Really happy to be on this panel and excited to learn from all of you about different aspects of this disease.
Thank you so much, Dr. Fauver. That was, can I double check that that's the correct pronunciation one more time? Okay. Okay. Yeah. So surprisingly, our first question was actually about what is COVID-19 and if we could talk about that. So thank you so much for that introduction. And I really want to say that we appreciate all of our panelists, how you're really trying to parse out some of these concepts that many of us haven't heard or know about, since we're coming from different fields of expertise. So we really appreciate you continuing to do that moving forward as we go through the questions. So I guess the first question, we'll just jump right into it. If there are any misconceptions about COVID that you'd like to address, myths that need busting, paradigms that have shifted for you throughout the pandemic. If anyone wants to jump in, or maybe Dr. Firestein, if you want to jump in here, you can.
Sure. I would say, well, maybe Dr. Fauver would like to comment on this more, but I think one that I've been hearing a lot recently is about testing positive after your infection. And I know that there's some confusion about the antigen testing versus PCR testing. And I think it's really important that as a scientific community, we're clarifying this. And I'm not an expert in virology or immunology, so I don't want to speak in an area that's not my expertise. But my understanding is that it is possible to test positive on PCR for several weeks, and in rare cases, several months after the infection. It's not clear that what's being detected is necessarily even live virus. RNA can, in some cases, sort of stick around for a period of time, but that doesn't mean that you actually have live virus circulating. Antigen tests, on the other hand, you really would not expect to be testing positive for very long after your infection, if at all. So those you can test more frequently than PCR. You may have heard it, if you tested positive, you can't do PCR testing again for 90 days, but you absolutely can and should continue to do rapid antigen testing even after an infection. That's one. And then maybe just one other, since so much of my focus has been on pregnancy and SARS-CoV-2 during pregnancy. Well, first off, there was early on a lot of hesitancy about pregnant women receiving vaccination. And I know one of the primary concerns was about fertility. And this has really, truly scientifically and medically been debunked. There is no evidence whatsoever to suggest that vaccination for SARS-CoV-2 is associated with any infertility or fertility-related issues. Fortunately now we're 2 years in, so we have accumulated a lot of data, and if something that profound were really occurring in our population, we really would know that at this point. So I would just like to clarify that, and of course, we now also know that pregnant women are at particular risk for severe disease if they are to get COVID-19. Of course, this is still a small percentage, but they are at risk. And so that's actually a population that really we're trying to encourage vaccination as much as possible. So those are two that have come up a lot in my work, but I would love to hear from the other panelists as well.
Well, I'll just speak again from personal experience because I'm not doing any active research. I'm involved in research as a patient, but I just want to clarify that I don't know if this myth is even going around anymore, but in the beginning it was that, you couldn't be sick long-term from COVID. Long COVID, what a lot of people with long COVID were kind of like gaslit for a while. saying that we must have had something else. I had even kind of lost friends over this who didn't believe me. And I just want to say that, you know, listen to your patients and what they're saying, especially with a new virus that people don't know, right? So I think that's something really important. And that, you know, unfortunately, long COVID is seen in the younger populations. From some of the studies I've read, they are seeing a lot of people in their kind of the prime of their life, their 30s, 40s, women mostly, but men are not, you know, unable to get it either. So yeah, I just kind of, I guess, wanted to clarify that. I don't know if anybody else has other research knowledge in this area, because again, I'm speaking more from a patient side, but yeah.
Yeah, I completely agree. I think the biggest myth that I feel like we've just been perpetually debunking is that COVID isn't serious, is that COVID is not really that consequential of a disease. And to me, what's been really interesting to see play out as you know, someone that was kind of in the space from a research standpoint from the beginning, when it did feel like kind of just a response to a pandemic, as this has just played out and it's gone, you know, we're over 2 years into this, we're still dealing with this in a really meaningful way, and people are clearly tired of it. What's been really interesting to see is I think kind of how COVID has shown so many different faults in our systems. And Ed Yong at the Atlantic's a really great science writer that has written these beautiful pieces about a lot of these topics. But you know, as Alyssa's talking about, you know, not being believed about a disease that surely doesn't just happen with long COVID, right? I mean, I think it's horribly problematic for people with long COVID, and something that's going to be with us for a very long time because it's so debilitating, and we've infected such a large percentage of the population with this virus that it's something we're going to be dealing with for decades. But that, you know, can also be reflected in other aspects of our healthcare system that I think, you know, COVID-19 has shown to be a problem. But surely, you know, it exists across these different systems. And in general, you know, this idea of, you know, kind of bringing it back to my work specifically, this idea of not pursuing mitigation strategies, not having flexible mitigation strategies, you know, trying to find a spot where, you know, everything in your life doesn't revolve around COVID, but being able to look outside your window and say, we're seeing increases in cases, maybe we should change what we're doing, you know, collectively, and have folks be willing to do that has been really, really difficult because, you know, Tony Fauci said this really early on with the variants, and I think it was like a really kind of nice, simple way to think about this is new variants don't show up if people aren't infected with the virus. So the best way to stop Omicron from sweeping through our population and whatever the next variant of concern that might currently be circulating we don't know about yet is to mitigate transmission. And you know, I'm currently in Nebraska now, where I'm originally from, after spending, you know, about 5 years out on the East Coast. And you know, there is no mitigation strategy anymore. We have more cases per day right now than we did during the Delta wave last summer, and there's zero mitigation strategies. And nationally and federally, kind of collectively, there is next to no mitigation strategies anymore. So this idea of just letting the virus circulate through the population because of that kind of original myth that like COVID's really not that bad is I think just going to, I feel like we're kind of perpetually stepping on brakes, and it's something we're just going to be dealing with for a very long time because of it.
Absolutely. Thank you guys so much for this. It really, I love that we are able to have these experts here and being able to talk about this from all of these different angles. And I want to just say that part of why I love this space so much, and I know a lot of our community does too, is because we finally get to talk to people that, you know what's happening and are able to share it with us in this accessible way. And it's sad, but it doesn't seem like those opportunities present themselves very often. I remember having conversations with my friends about the new CDC 5-day, 10-day rule. And even though we're in, teachers college, we're studying neuroscience and education, it was still this questioning of, how do we really understand these papers? Who do we listen to here? What are the, data sources we want to really trust in this case? And so just reminding me how important this discussion is. Sorry for the little tangent, but I wanted to highlight a question from the chat. Can we learn more about what specifically happens to the brain during and after infection? Feel free to jump in, anyone who wants to start, and we can kind of take it from there.
That is very specifically outside of my area of expertise, so I'm going to bow out for that.
I mean, I will say that I think it's one of the current sort of million-dollar questions. We don't know very much. I mean, our group right now, we're actively working, as I mentioned very quickly, we're doing MRI and fMRI scans of mother-infant dyads. So what we're hoping to learn from that is both, are there any structural differences, both of the person, the mother who had SARS-CoV-2 while she was pregnant, but then also are there other effects that might have reached the fetal brain during development, or even are there postnatal effects? So we'll be looking for structural differences, but also what we call functional differences. So we can actually look along different pathways that different brain regions might interact with one another. For those who are involved in this area at all, we're mostly looking at prefrontal regions and the amygdala, which is really important for a lot of regulation, emotion regulation, sort of higher order executive functioning type tasks. So I'm, really excited to see what we learn there. There is a paper that came out not too long ago showing that they actually had this wonderful cohort of patients who had had an MRI conducted for research prior to the pandemic. And then again after, or I'm sorry, I'm not sure if it was before the pandemic, but it was before they had an infection. And then again after they had an infection. And there was some suggestion in the data that there might have been a decrease in the gray matter of those patients if they had an infection compared to those who did not. But I think, that type of work we really need. It is a quite robust and large study, but I do think we need replication and really looking at different, the spectrum of severity of disease. Does that make a difference? So I think at this point, it's a little bit early to really give a reliable answer to that question, but it's a great one. And definitely a lot of people are working on trying to answer it.
Yes, I would agree. I also don't have an answer to that question. I think it's currently being researched. I can only comment on individuals I've worked with and patients that I've worked with. The neurological symptoms are very real. People complaining of brain fog, tremors, concentration issues, memory issues, cognitive fatigue. I've worked with patients that have had strokes while having COVID, and they're not sure. That's probably due to blood clotting issues or pulmonary issues. But I think that question is a really pertinent question, and it's currently being researched. So hopefully we'll have an answer eventually. Great question, though.
I think one thing to maybe add on to this kind of more broadly is that COVID-19, as the disease, an infection with SARS-CoV-2 varies drastically in the population. It varies by immune background, whether or not you've been vaccinated beforehand, before your primary infection or not, what variant circulated at the time, and then from person to person. The likelihood of being infected with the virus varies drastically, but the clinical presentations, it would be interesting to hear from a clinician that's kind of worked on this. But you can hear anecdotally. If someone's like, "Oh yeah, I had COVID and I didn't even know it. I tested positive because I had to, take a rapid test for work, and I had absolutely no idea, and I had no symptoms." But then you have other folks that it's incredibly debilitating for. So as far as COVID-19's effect on the brain, I'm sure folks looking at their answer would be it varies. It's going to differ from individual to individual. It's in all likelihood some very complex combination of the virus genotype, human genotypes, risk backgrounds, bad luck of a variety of different instances are likely going to change that.
Totally. And I think the question is kind of, in some way hopeful, right? We want to know so much about this, and we're kind of hoping that the research is maybe further along than it is, or that there's some sort of clear-cut answer. And unfortunately, it just doesn't seem like we're there. But I kind of wanted to pick your brains and see if anyone had kind of read anything about that. But I can pass over to Sarah to move on to the next question.
Yeah, thank you, Mahima. This one is directed toward Dr. Pacheco. Are there any lessons that you've learned from your work that you apply to your daily life, or that you recommend that our audience applies? And I know you spoke a little bit of this to this in your introduction, but we'd love to hear more of your insight on that.
Yeah, of course, that's a great question. I think the biggest thing for me was fatigue management. I've kind of applied personally to myself while I was healing and getting better, this kind of like point system that was actually developed initially for people with concussion to prevent overstimulation and like cognitive fatigue. So certain things have a certain amount of points, kind of like Weight Watchers. Watching TV might take 2 cognitive points, or making a meal might take 4 or 5. So I kind of like had to find my threshold of how many points I could tolerate in a day and plan my day around that. So I knew if I had a class, because I was still in school, I had to kind of plan my day around that class and make sure that maybe I had made dinner the night before that I would heat up that night, because that would take less energy. And as I improved in time, I was able to tolerate more points a day. And sometimes if I overdid it, I would kind of backtrack for a day. So I've actually used that strategy a lot with my patients now going forward, not just with long COVID, but chronic fatigue syndrome, anything they're really healing from where it's taxing on their body. And it's been really helpful for me. And the patients that I've worked with and the clients that I've worked with have said it's been helpful to them also. So that's something that maybe others could use going forward.
Thanks so much for sharing that. I think many of us here are either clinicians or educators, or work with people that are clinicians, or are just wanting to treat people the right way that we're around, that we know that are experiencing this. So thank you so much for sharing that. Mahima, I'll pass it back to you.
Totally. Maybe this is a broad question, but I found it kind of interesting. What is something that surprised you about COVID-19? Something that you really didn't expect and kind of, took you a second to process?
Well, I can say, so the data that I showed in my introduction, so if you remember, we first had our 2 groups of babies who had been exposed to SARS-CoV-2 in utero and those that had not. And that was going to be our paper. We were, ready to send in this totally negative finding, but it was a great negative finding. Can't say that often in science, but we were really excited. And at the last second, we were like, you know what, we actually have this historical group of infants, like I mentioned, who were all born and assessed prior to the pandemic. Let's just throw them in as a 3rd group, just to really show there's no difference. It's sort of like an extra control group, because we were treating our unexposed infants as our original control group. And we were like, well, let's just take it one step further and have another control group. So being completely transparent, that was not our original plan for the paper. And it was at that moment, I will never forget it. I think it was 9:30 at night. I was in my room alone working on this analysis. And that's when I saw this big effect, that babies born during the pandemic were scoring significantly lower on some of those developmental domains compared to the babies born before the pandemic. And I think, if there are any scientists on the call, you probably have, or I hope at some point you do have the experience of realizing that you know something that probably nobody else in the world knows quite yet. And it was incredible. I immediately called Danny Dimitriou, who is the lead PI. She's a well baby pediatrician, and she's really been running the combo study. And we sort of just sat there on the phone together, just over and over again saying, oh my God, what does this mean? So, it's not what we thought the data would show. It was definitely a surprise, but being, from her perspective, a pediatrician, from my perspective, a developmental psychologist, this is exactly the kind of information that we need to be getting and looking at so that we can, come to people like you who are working with families and working with patients. And really you can then have informed approaches when referring patients or providing different services to them. So it was a surprise. I'm still not sure if it was a good or a bad surprise, but hopefully it's a helpful one.
Do another panel? Yeah, go for it. Sorry.
No, I was just gonna say, I think just for me was was realizing that COVID could be, severe, and you didn't have to have a quote unquote severe course for it to have long-term effects. I've unfortunately worked with many people, both in acute care and home care, that have, are still pretty debilitated and have a lot of difficulty functioning from the virus. Unfortunately, I've had people that have, been vaccinated and boosted that have had the same course. I know that's probably not what people want to hear, but I think we just still need to take protective measures to protect ourselves as best we can when we know that there's waves coming. Whether that's social distancing or masking up when we're indoors. I think it's just really important that, the vaccines and boosting obviously protect us, and we're so grateful that those were developed, but I still think mask wearing is still very important for the waves that we have to protect us, because we don't know how sick we'll be until we have the virus, and to try to avoid that at all costs, I think is really important, personally.
Yeah, it really is. Thank you all. I have actually a little bit of a follow-up question for you, Dr. Firestein. And this is kind of an open question. Anybody can answer this too. And it's one of those $1 million questions, but I just want to pick your brain about it. When you're seeing these results and these effects on babies and their cognitive functioning, whether or not their mom had COVID or not, I'm wondering if you're thinking that the stress of the mother during the pandemic, of living through a pandemic, even as a pregnant mother, like even as a woman who's pregnant, how would you think that that's part of the reasoning, or is there anything else that you're kind of attributing to it, like not being able to communicate as much maybe with many people? Just wanted to hear your thoughts on that.
Yeah, so I would say there's sort of kind of 3 primary theories that we're looking into right now, because we've, first off, let me say, we did one study. Please, if anyone has the ability to try to replicate our results, we are, hoping to see that soon. So I can only speak about our results. I hope that others will follow, but we sort of have 3 major areas that we're looking into now. So because we don't currently see an effect of the virus. So definitely stress is one. When we talk about stress, we're sort of thinking about stress in very broad terms. So we do get measures of, maternal reported depression, anxiety, specific changes because of COVID. So, especially early on in the pandemic, prenatal visits were shifted to entirely remote. A lot of women had delayed scans and visits with their doctors. Their birthing plans had to change. If anyone remembers, in New York City in the very, very beginning of the pandemic, partners were not even allowed. So women were, in labor and giving birth completely alone. Actually, one of my colleagues and several friends of mine were in that situation. And I think it's still something that they're processing and experiencing. So fortunately, that practice has not continued. But anyway, so we include stress really broadly. Unfortunately, just because of the nature of science, getting IRB protocols up and running and getting our surveys and study out to the participants, we don't have measures of maternal stress during pregnancy for those women who were pregnant during the very peak of the pandemic. So that's one of our biggest limitations. We can never, go back in time and get that data. But we do, moving forward, now have stress measures during pregnancy and throughout the first year postpartum. We also think, there's some literature out there that stress, especially during the postpartum period, can actually affect parental perception of behaviors of their children. So it's quite possible that there actually are no developmental differences, but the ways that parents are interpreting certain behaviors or just being home more with their young infants and being able to see the behaviors more might sort of, be shifting how they're reporting about infant development. And then the third is, the infants themselves are having a very unique early postnatal experience. They're, like you mentioned, they're not meeting as many novel people. I mean, now maybe things are starting to change a bit, but when we think about first wave, these babies were mostly probably at home with the same, one to few caregivers. And, a lot of families were not even taking their infants to the grocery store for the first year of their life. So just a very unique experience. And I think there are a lot of factors that, it might not be one as well. It might be a convergence of all three of these, for example. So those are the sorts of things that we're currently thinking about.
Thanks so much. Just check if anybody else wanted to jump in on that one.
Oh, sorry. Can I just add something? Because I see Leary put a great comment in the chat, sort of bringing in the immune aspect. So that's another thing actually that we're trying to get at. So there's this concept of maternal immune activation, which definitely, during an infection, the immune system activates. But interestingly, even stress can be enough to trigger the release of certain immune markers that we, for example, cytokines. So we're measuring cytokine levels during pregnancy. We're trying to do some even postpartum, perhaps. And so it's possible that those sorts of immune markers could even be affecting the fetal brain. There's actually quite a large literature on maternal immune activation and infant development.
If I could, I wasn't going to hop in on this panel, but I got very excited because you are all speaking so incredibly, specifically to what Dr. Firestein just said. She was talking about how different elements may each impact, be it internal factors in the infant, be it the interaction with the caregivers or the larger environment. And that is absolutely true, but it will absolutely be all three and even more. Right, so it's not even about trying to understand which of these things has most impact, but understanding that everything is going to have an impact. So thank you for that.
Yeah, thank you.
Yeah, thanks, Larry. Such a good point. Sometimes we really want to just have solutions, right? But it's a really tough time that we're living in that we just have to really understand that there's so many factors at play and we still have a lot to learn. Dr. Fauver, I wanted to ask you if there's anything that surprised you about COVID-19, if there's anything that comes to mind that you'd like to add, but no question.
Sure, yeah. No, it's a really good question. And it's hard to not get existential about it, right? Because the last 2.5 years has been just so, it's been so different. It's been so hard for so many different reasons, for so many different people. And I've found it, personally, I found it difficult because there was never really any way to disconnect from it because we were doing the work and we come home and have to deal with I had a daughter born in December of 2019, so really interesting to hear about the effects on children. For an N of 1, yes, she stayed home a lot. We went to the grocery store for the first time like a few weeks ago. But as far as like scientific findings that were surprising, I actually got one of the first papers we published on it, we thought was really interesting, where we sequenced the virus from the first patients in Connecticut. And if you remember, kind of back in early 2020, there's reports of this new respiratory infection caused by this novel coronavirus in Wuhan, China, and then you start to see other countries see it. And that's, I think, where a lot of folks that have kind of been in this space for a while knew that at that point, like, okay, this containment is that ship is actively sailing. And then the first cases in the US were in Washington kind of in February. And then there was this like first 2 weeks in March where it was just like where it was first, just like every day it was like, this state just identified their first case, this state just identified their first case. And then we were in Connecticut, and I think our first case was on like March 6th. And then because they were diagnosed at Yale New Haven Hospital, we were able to actually get samples and do this next generation sequencing approach, and then do what's called phylogenetics, basically to ask the question, how are these samples related to each other, and then all of the other samples that we know exist. So kind of that scientific moment of something that no one else does, that Dr. Firestein talked about, happened to us when we sequenced these viruses. It was like 3 in the morning. We were looking at the trees, making a phylogenetic tree, which is basically, again, just to say like, how are these viruses related in space and time. We actually found that the ones from Connecticut, because the common sentiment at the time was these were all being imported from elsewhere in the world. There wasn't community transmission. The viruses we found in Connecticut were most closely related to the viruses identified in Washington about a month before that. And so kind of the takeaway from that broadly was, I think, something we already knew. Of course there was community transmission occurring. This was absolutely happening, but it was one, we didn't have testing capabilities. There was a big kind of testing kerfuffle at the beginning of this, where all the samples that we sent to the CDC, the assays they're using weren't working very well. You had to have like international travel or close contact with a diagnosed case, and like if they can't get their cases diagnosed, no one's going to have close contact with a diagnosed case. And our data, along with a lot of other folks, this was by no means like the definitive paper on this, suggested that throughout February, early March, the really early parts of the pandemic, there's absolutely undetected community transmission occurring. And we could demonstrate that not only from a testing standpoint, but using the virus genomes themselves. What else was more surprising? The thing that surprised me more broadly about all of this is the variants. As someone that studies viral evolution, virus evolution often wants to get reduced to like this mutation and this gene results in this phenotype, and it very rarely is that the case. I think with even like genetic diseases in humans, that's not nearly as clear cut as we would like it to be. So when the virus at the beginning, the virus, when the virus transmits, it has to replicate its genome. The replication machinery is error prone. Mutations happen, and it happens in a clock-like fashion. The mutations themselves are not unexpected. We always expect, especially RNA viruses, to mutate. The fact that they've mutated in such a way that has made, so Omicron, the virus we're dealing with right now, I think reasonable people could make an argument that this is potentially the most infectious disease, at least respiratory disease, that we've ever seen. So something that went, was very infectious at the beginning, that by no means was a slouch at infecting people, to what we have now, due to less than about 100 mutations across the viral genome. That's been just broadly very surprising to me, that we keep seeing these changes. We're seeing immune invasion directly due to mutations in the viral genome. And maybe in retrospect, when I didn't think it was going to be at the scale that it was. It was a global pandemic perpetually. When you have millions of people at any given time infected, random things aren't that random anymore, right? When your N is that high, you can expect these things to happen. So I think looking back on it, and like, you're like, well, this is how it's going to play out. I'd be like, okay, yeah, no, surely we're going to see evolutionary pressures on the virus to change. But if you would have told me this at the time, I would be like, no, mutations probably don't have that much to do with the outcomes here.
No, that makes sense, and it's so interesting. I know you talked a little in your intro about the vaccine and its development, but I'm wondering if we can kind of follow up on that and as it relates to all the variants. But it'd be helpful if you can give us kind of an intro, imagining that we know absolutely nothing about vaccine development, because I'm sure a lot of us are, including me, a little unfamiliar with it. Kind of how is it developed and how does it help to fight COVID, in terms of, maybe if you have a perspective on like long COVID, on how it, works with all the different variants over time, things like that. We'd love to hear your expertise on that.
Yeah, sure. I can maybe kind of give a quick overview. So I'm also not a vaccinologist. There's some really great, actually at Columbia, there's some incredible vaccine people. But in general, so the big thing was the mRNA vaccines, right? That's been the big difference. That's, I think, kind of allowed for the vaccine to be developed as quick as it was and to get through trials as quickly as it did. So like Pfizer and Moderna are the most common vaccines, and what that is just a piece of the RNA. So let me actually just pull a slide up again. I think that potentially could be helpful. Visualization cartoons is like what I need to understand molecular biology. I don't know if I'm the only one in that boat. So the vaccine is made to the spike protein right here. So it's not the entire virus. So this is an RNA virus. So it starts here, it ends here, and when you get infected, it just replicates itself over and over and over again, right? So what the mRNA vaccines did was take that gene sequence for just the spike protein. That's the protruding parts here. That's coronaviridae. They look like little crowns. That's the idea.
Excuse me, I don't know if it's just me, but I can't see your screen.
Oh shoot! Sorry. Can you see it now?
Yes. Okay.
Apologies. So whole genome starts here, ends here. This spike protein is what the vaccines are made to. It's these little bits here. It's called coronavirus because the spikes make them look like a crown. But this is what's most immunoreactive. This is what your body, the antibodies will recognize because it's kind of on the outside of the virus. So the mRNA vaccines are just the RNA sequence of the spike protein. So it's a new vaccine technology for sure. And part of the advantage, at least as it's being sold, is you can change that sequence relatively quickly. So the spike gene, so this protein sequence for the virus that was first detected in Wuhan, is what that vaccine is made to. That spike gene's changed quite a bit. It's how consequential are any of the different mutations. It's varied, but it's not the same thing anymore. But there's still really good evidence to show, especially as far as protection against severe disease, hospitalization, the worst outcomes of SARS-CoV-2, death. The mRNA vaccine derived from that original spike protein sequence is still very, especially from a booster standpoint, is still holding up very well. There's a variety of other vaccines. Novavax just recently got approved in the last week or two, which is more of a traditional protein-based adjuvant vaccine. And similar to like the flu shot, it's not developed exactly the same way, but it's still like kind of a protein-based vaccine. So as far as how that relates to variants, there's ways to update them. That's part of the big benefit of the mRNA vaccine. There's a trial that just went through where they have been updating it with both the original vaccine strain and an Omicron version of the vaccine strain. And that's shown to be, I think, more protective against infection. But just like the original vaccine trials, you've got to go through, there's no cutting corners there, right? You have to go through all the regulatory processes, the phase 3 clinical trials, and do the independent assessments. Any new update to that vaccine, for the most part, will have to follow that same path. So there will be time, and I think just kind of think about it in general, it's kind of hard to decide when to do that, right? Delta, if you'd have asked me 6 months ago, I'm like, yeah, Delta is the circulating variant. Everything's Delta, and anything new is going to come from that. And then Omicron is entirely separate. So the question is, will anything new come from Omicron? And what we're seeing now the nomenclature, I know that can be kind of difficult sometimes, like what's BA1 versus BA2 versus BA4 and BA5. They're all kind of different flavors of Omicron. So I think they've decided to move forward with this bivalent Omicron sequence in a vaccine. But there's no promise that that's going to be the circulating variant 6 months from now, when potentially it could get approved. And as far as vaccines and long COVID go, I've heard most recently that they're not terribly protective. Dr. Pacheco just mentioned that, and I'm sure she would have more information on being vaccinated and boosted and potential whatever potential protection that might provide against long COVID.
It's a great question, and I know many people are curious and like how they can protect themselves the most from long COVID, and I don't think we really know the answer to that yet. From my experience working in acute care, the vaccines absolutely have kept our hospital system from being overrun during these upswings. They have seemed to keep people out of the hospital for long periods of time, and they have also kept people from death, which is the biggest and the main thing that we're obviously trying to avoid. So the vaccines are doing their job. I mean, yes, people are still getting sick, and I've still worked with people that are getting long COVID and they've had the vaccine and been boosted. I wish I had a better way to say how to avoid that. I can say from my personal experience, obviously the vaccines were not around when I had COVID. But once they finally did blood work, they had seen that I was extremely low in vitamin D. I know that was something that was going around as, could that cause people to be more sick? They also found that I was very low in something called glutathione, and I actually had to get infusions of it. Again, I don't know from a research standpoint if those things can help protect you from long COVID. I would actually be interested in going into the literature and seeing if they are. But yeah, that's about as much as I can offer. I'm not sure if any other panelists have a say on that or know more about the research in that area.
That's okay. Yeah, thank you so much both for sharing your insight. We can move on to our next question. And we got this question a lot. It was one of our most highly requested topics about why there's so much variation across groups. So age, race and ethnicity, socioeconomic status, and other factors. And this is a tough topic, right? But it's a really important topic. We know that systemic inequity and just trusts in the vaccines themselves, but also in the healthcare system across like all of America, right? So we wanted to ask, what do we know about the effects of COVID-19 on different groups? If you've come across any research that struck you in that area, or do you have any hypotheses at this point for why such variation exists? I'll give you all a moment to consider.
I think I could maybe touch on this just broadly first, and then let the other panelists, dive in. So most of my work prior to COVID was on what's called neglected tropical diseases, or also known as like diseases of poverty. And I think what might be worth pointing out to start from this discussion is impoverished people living in poverty almost inevitably bear the brunt of infectious diseases, especially in 2022. Infectious diseases, I think, 100 years ago before that, were potentially more widespread, but for a lot of these really consequential infectious diseases, it is almost inevitably going to be worse for people that are less resourced. And I think that's potentially where the starting point for something like this needs to be, of why it's affected some communities worse than others, and thinking about it not just in the United States, but elsewhere across the world.
I can also maybe speak broadly about this. I'm involved in in a research project at Teachers College where we're looking more into racism and, you know, the effects of some of those things on healthcare. So this is again from not necessarily a COVID perspective, but I know the literature has shown that there is a lot of mistrust in the healthcare community for Black and Brown people, dating back from when research was done unethically on individuals of color. And unfortunately, that has caused a lot of mistrust and less involvement in research. So I think maybe this is just my opinion, flashing forward to now with vaccines for COVID and maybe why certain people aren't getting vaccinated, because we know it's not necessarily that they're not being offered the vaccines. I think it's still maybe related to mistrust or knowing that maybe the vaccines weren't tested on someone like them, and that can cause fear for some people. So I think it's can be related to that. I think it's also, you know, it's a difficult question and difficult, it's difficult to pinpoint. I think it's a very, you know, real problem that needs to be addressed. I'd be interested to see if anybody else has any other comments, but that's my take on it.
Yeah, I mean, this is not my area of expertise. I'll just say that so Danny Dimitriou, the pediatrician who's really been leading Combo, was really spot on and had the foresight and early recognition of the fact that you know, the burden of SARS-CoV-2 is really just superimposed on these pre-existing, what we think of as like social determinants of health, for example, and these inequalities. And, you know, that certainly is not new. It's just, it's been exacerbated, I think, because of the pandemic, of course. And in all of our sample, so we do a matched case control design. So for every case of SARS-CoV-2, we then enroll dyads who are not exposed. And we match them on a lot of different things like the baby's sex and age at delivery and important variables that we know are predictive of long-term outcomes. But Dr. Dimitriou, I think, was spot on in recognizing that we actually could not match for things like race, ethnicity, different socioeconomic variables, because that would really, you know, it would negate the ecological reality of what we now understand has happened during this pandemic. So we do see in our data the exact type of inequalities and disparities that you're talking about. And I'll say our patient population is a really interesting one. It's highly diverse. This is all done at the Columbia Medical Center. About, depending on our subset for an analysis, about 50 to 75% of our sample self-identifies as Hispanic. So we do have a quite diverse sample, and it's something that we actually have many people on our team who focus on social determinants of health and health disparities. So if anyone on this call is particularly interested in that area, I would really encourage you to reach out to us, because we're looking for more people with expertise in that topic.
Yeah, I just want to add on, Dr. Firestein, that like as someone who's kind of connected to Combo through my lab, not directly, but it's been so inspiring to see, I think it's like 70 different professionals that are involved on this project and all coming together to address these issues and really, really consider more than just what is typically in our own research box. So I just wanted to kind of give a little shout out to Combo. And thank you so much for your work on it.
Oh,
what was that?
I was just working with people from all these different areas. It's been to my benefit. I mean, I've learned so much that I really, you know, should have known before and I didn't. So it's been a great experience.
Yeah, and I'm hoping that, you know, Dr. Froud mentioned the silver lining of the Synapse coming together, and I'm hoping that a silver lining of this can be just more of a collaborative research approach in general in the scientific community. So let's hope for that. All right, I'm going to pass it to you, Mahima.
Amazing. I know we're kind of getting close to the end of our time together today, and I kind of wanted to open the floor really to the three of you. I know we've talked about a couple different things and jumped to a couple different places. Sorry, it seems like there's some kids outside. Kind of as we're finishing up, we wanted to see if we can move the conversation to what you all are kind of hopeful for or optimistic about as we're kind of moving through. I know we're not at the end of the pandemic, but being in the spaces that you are, and with your areas of expertise and your kind of even like personal feelings about this, kind of where are you and where do you see us going from here? And I feel like that might be a good way to kind of close us out today.
I can jump in. So from my perspective as a clinician, occupational therapist, I would really like to see more studies on like the effects of physical activity for individuals with long COVID and post-COVID syndrome, and where they're starting from, perhaps before getting COVID, during COVID, after COVID, so that we can best find a way to help them get back to their daily lives and their physical activity that they were doing beforehand. So that's one thing. I would also like to see, obviously a cure for long COVID, for there's a lot of people that are still struggling, that don't have answers, that have gone through therapy and still feel at a loss. And I think there needs to be a lot more research in that area. I mean, at this point, there's probably millions. I think I last read anywhere from like 7 to like 15 million people struggling with long COVID. They really don't know the numbers exactly. And I think that in itself is going to create kind of its own little mini pandemic. A lot of people are going to need help in this area. A lot of people are going to need therapy, counseling, physical therapy, occupational therapy, medical needs. And I think that's something that needs to be addressed socially and via healthcare. These people might be out of work for a while. I know I was out of work for over a year. How are we gonna fund that? There's, I think, a lot that needs to be thought about from that sense. So I hope that some funding is allocated for that purpose going forward. And thank you so much for having me. This was a wonderful discussion.
We're really so happy to have had you here today, really.
Yeah, I would just piggyback off of that and add, it is somewhat encouraging that the NIH has sort of funded this big initiative called, it's called PASC. I think it stands for post-acute sequelae. I can't remember exactly, but it's really targeted at understanding long COVID. And so, I hope from a research perspective that there be, more data does become available and that can guide care. But in the meantime, I'm really encouraged to know that there are clinicians like you, Alissa, that are working on this and trying to help patients. So thank you for your work in this area. I would say, from my perspective, I hope that there is long-term follow-up of these kids and mother-child dyads and parents and families that have gone through the pandemic, regardless of if anyone was infected with SARS-CoV-2, really understanding the long-term impacts on child wellness, health, development. So that's something that we'll be working towards, and I hope others will join us in that. And then I think the last thing is really just recognizing sort of as a community what we've gone through. And, I always find this interesting as one part of our study, it's a survey that we give out and we ask women, it's a very long survey. It takes about 45 minutes. And at the very, very end, there are 2 optional questions. What has been the most stressful thing for you? And do you have any advice for other pregnant women or new parents that you'd like to share. And I just find it really powerful that typically about 75% of the participants take the time to complete a completely optional question. And they share really insightful experiences and advice. And I just hope that sort of a community comes of this, and that has some impact on those that are currently going through it and in the future. So I'll leave it at that. And just again, thank you so much to the organizers of this. I've so enjoyed being part of it. And to the other panelists, I've really learned a lot from both of you. So thanks so much for your thoughts today.
Yeah, no, I second all those sentiments. I've learned a ton from both of the other panels here, and then some incredible discussions in the chat and questions. And so as far as what am I optimistic? I feel like you kind of have to preface all of this. I don't know if there's definitely things to be optimistic about, but there's also things that if we pretend they're just not still happening, we're going to be dealing with those consequences for a long time. So I do feel like it's really up and down. I would say, like, kind of broadly, what can we be optimistic about is, I would say, to summarize it as quickly as possible, past infections don't count for nothing. We're seeing this kind of decoupling effect, and I feel like there's been pundits have been saying we're going to see this from day one, and of course that hasn't been the case. But kind of the latest data out of South Africa, where the newer Omicron variants have been circulating, the BA4 or BA5, if you're up on the lineage nomenclature. They did see an increase in cases, but as far as deaths over expected at that time period, which I think is kind of our most reliable metric of that outcome, they're not really elevated, which I think the simplest and most judicious reading of that would be like, yeah, these new variants might continue to cause more cases, but they hopefully are not going to cause as much severe disease, hospitalization, and death. But as Dr. Pacheco's shown us from her personal experience, that isn't the only bad outcome, right? There are other outcomes of COVID that we need to consider when we're looking at case rates like long COVID. So I think we're going to see less people dying, which is a good thing, but if we still continue to see just unabated transmission, we are going to be dealing with all these other consequences associated with COVID infections. And broadly, I think we all in this space kind of feared this, and I think it's kind of actually happening. It was mentioned in this chat earlier on, is money's already starting to be reallocated from the COVID response to other things, whether that be from like a purely NIH funding standpoint, that money that would potentially be going to like state and local health departments might be going now to the vaccine rollouts and things getting reallocated there. But also the kind of social support networks that were put in place because of COVID. I'm thinking about like the money that went to people with kids. Just I'm not going to pretend to be like a politician here, but that came in one of the most recent COVID bills, and it just gave money to folks with kids, and it helped alleviate some of these financial hardships that a lot of folks were going through because of the pandemic, and like that's expiring soon. So I'm concerned that we won't learn our lessons, that we will have a bit of a we've kind of already had a short-sighted pandemic response. But I would love to paint like a rosy picture that like we all kind of coalesce together around the common good of COVID. That obviously just didn't happen. And as someone that works in a variety of different infectious disease systems, this will not be the last pandemic. There will be something different. There will perpetually be something different. COVID always kind of seemed to me as like this perfect combination of infectiousness, and it wasn't so deadly that like it wasn't to compare it to other infectious disease systems that they could have like really high infection mortality ratios would be very obvious, like you can't do it because from the very beginning I think kind of bad actors were like, oh look, it's really not that bad. I kind of mentioned that at the beginning. So will we see something like COVID again? I don't know. I'm not going to pontificate on that, but pandemics of long forgotten or new infectious diseases are just going to keep happening. So what I want to be optimistic about is the kind of these communities coming together, the scientific communities as well as policymakers, recognizing the importance of these things, recognizing that prevention is absolutely the way to go here, and putting in and reinforcing mitigation strategies and taking lessons that we've hopefully learned from this pandemic and applying them more broadly. That would be something I'm hopeful for.
Yeah.
Thank you both. I also just wanted to thank both Dr. Firestein and Dr. Fauver for all of your insights. And it's been such an honor to be on a panel with both of you. Me still being towards the end of my doctoral schooling. So I just wanted to thank you both for this opportunity.
Amazing. Yeah, thank you all for sharing your closing thoughts. And I also just want to really encourage us all to, look at the chat and consider what's the really important points that have been raised there. Things like misinformation, we talked about Tuskegee, all of these. I mean, that experiment it's decades and decades of impact, and it's going to continue to have that impact. But I really want to thank you all for raising and speaking about these really, really important topics in the chat too. And I encourage us all to take a look at those. And with that, I also want to thank all of our amazing panelists. Can we all thank them in whatever way we feel comfortable with in the chat? Give some reactions on Zoom, whatever you want to do. I think I can speak for all of us when I say that this has been so informative and empowering. And I really, really hope that somehow this information can be disseminated more widely and that we can all as a society and culturally just do a better job of that in whatever way we can, big and small. And yeah, so I just want to thank you all for such a thought-provoking and informative discussion. And I think all of us feel so strongly at the Synapse that these multidisciplinary conversations, such as this one, are a really important way to improve our approach and to move forward and heal. And so with that, I will say we're going to take a brain break until our next panel, where we will be diving into the impacts of COVID as a pandemic rather than as a virus. We've all experienced and witnessed, as we've been speaking about, the impacts on areas such as mental health, sociology, economics, physical well-being, whether or not we've gotten sick ourselves or seen others become ill. And coming from this panel that looked more specifically to viruses and disease, will be broadening our view and looking at its impact on our daily lives. The speakers for our next panel are Dr. Kate Havens, who I saw in here earlier. Thank you so much for being here. Dr. Cassie Hendricks, Dr. Doya Arad. And this panel will be moderated by our very own Teachers College Neuroscience and Education Program Director, Dr. Karen Froud. So with that, we want to thank you all again so much. We look forward to seeing you at 3 o'clock. The Zoom link is on our Eventbrite link in your inbox. And in case you still can't find them, they're also on our Instagram. And I think Mahima is going to show that now. Thank you all so much, everyone.
Thank you so much for joining us.